Students, welcome to lecture 27 in biopharmaceutics and pharmacokinetics, and we are discussing chapter pharmacokinetics of intravenous infusions. In that series of lectures, today's it is the last lecture, and it is intravenous infusions and 4. And in this one, we will discuss about the clinical significance of pharmacokinetics of intravenous infusions. Outline for today's lecture. Um, Clinical significance of pharmacokinetics of intravenous infusions. Till now, we have discussed that what are intravenous infusions and what is steady state level, how long it takes to calculate to achieve the steady state level, how it can be calculated. Now we are discussing the same thing from clinical significance aspect. So to achieve the steady state levels fastly, how it can be incorporated with the loading doses and the steady state levels can be at which are the desirable states fastly to avoid potentially dangerous situations. Then pharmacokinetics of the drugs which are following two compartmental models and to avoid the toxic concentrations. So these are the main applications of clinical applications or significant points of pharmacokinetics of intravenous infusions. So clinical significance of pharmacokinetics of intravenous infusions. Intravenous infusions have a whole lot of significance related to their use and the various situations where the intravenous infusions are adopted. For example, intravenous infusions are particularly helpful in achieving steady state levels immediately, which is very desirable. Um, though in previous lecture we have discussed that no matter whatever you try, steady state level would be achieved at a certain time period, but um, it doesn't true. It is not true. It doesn't hold true, or it is not true in a situation where you are administering intravenous infusion along with another um, dose, which is also called the loading dose. And in that situation, the intravenous infusions can be helpful in achieving the steady state levels immediately. To avoid potentially dangerous health situations, as we discussed already, that there are drugs which got low therapeutic indices, uh, they got narrow therapeutic windows, and it is very easy for such drugs to cross a toxic concentration within no time. Examples are digoxin, lithium, and gentamicin. And that's the reason that most often for such drugs, therapeutic drug monitoring is suggested. The main reason is the same that as it is desirable to observe whether the patient is experiencing the therapeutic benefits and the toxic effects or the side effects are just compromisable and not crossing the dangerous limits. So that's the main reason that intravenous infusions are adopted in such situations. Then the next important benefit is to avoid toxic concentration with the help of intravenous infusions. As we had discussed already, that uh, the moment you stop intravenous infusion, the moment it drops, the curve drops exponentially. There is an exponential decrease in the plasma drug concentrations, and that avoids any further increase in the concentration or to be maintained at that level which was creating the toxic effects. So it is helpful to avoid the toxic concentrations as well. So these are the various factors which are specifically associated with the pharmacokinetics of intravenous infusions and which make them so desirable. So one of the frequent use of intravenous infusions is to deliver loading dose rapidly, and one of the way or the popular method is to administer the loading dose and intravenous infusions concurrently. Now coming to the point, this thing seems in contrast to what we discussed in our previous lecture, that no matter how much you double the intravenous infusion rate, the time to reach the steady state level will always remain the same. Now, the fact is that that thing is still true. But the point is that you are administering the intravenous infusion along with another loading dose, which is intravenously being administered. That is, which one is the intravenous bolus administration. And so that loading dose will help to build up that concentration within the blood, uh, which is higher and which is just equivalent to the rate of intravenous infusion. So what happens that when you start the intravenous infusion along with the intravenous bolus loading dose concurrently, intravenous bolus dose helps to, or the loading dose, simply you call it, it helps to attain that concentration within the blood, which brings the rate of elimination of the intravenous infusion just equivalent to the rate of infusion more rapidly. So that is the main reason that you attain the steady state level uh, more immediately or more rapidly, and which is not possible if you are administering the intravenous infusion alone, or though you are increasing, you are doubling the rate, but still, though the dose is being administered at zero with zero order, it is being eliminated from the body with first order. First order means that the elimination rate is dependent upon the drug concentration within the systemic circulation. So there is a trick basically. With the help of the intravenous loading dose bolus administration, what you are doing that you are providing that already built up concentration within the blood or the systemic circulation, which will be mistaken by the elimination rate of the rate of infusions administration uh, as the drug which is already present in the body, and so it will speedily build up and come to the level of the intravenous rate of intravenous infusion, and soon the rate of input will become equal to the rate of output, and that is the main reason that steady state level is achieved earlier in this situation. In this, steady state level is achieved almost immediately. Okay, so coming to this graph, the discussion which we made in the previous slide, this graph explains that, and in that we were discussing that if intravenous infusion is concurrently administered with a bolus administration, what will happen that the steady state level can be attained immediately, almost immediately. So you can see that there are various possibilities. The bolus administration or the loading doses could be of various concentrations. But anyways, the point is that they help to build up the concentration in the blood, which helps to bring the rate of elimination, which is um, in case of intravenous infusions is following first order, it helps to bring that more rapidly equivalent to the rate of infusion. This concentration, which is built up already by the bolus administration or the loading dose here in all the three cases, A, B, C, just the differences of the dose which is being administered. C is lesser higher, B is this much, and A is almost very higher dose. And but whatever, finally it comes equal to in which a point where the rate of infusion becomes equal to the rate of output, and we achieve the steady state level. So if let's suppose it is the normal situation, what will happen that it will take for sufficiently longer period for intravenous infusion to achieve the steady state level, like in this way that for example, let's suppose these all are missing, these all parts are missing, we are not administering any additional loading dose, only the intravenous administration infusion administration. So it is a possibility that it will take sufficiently longer to achieve the steady state level, or maybe like this. But in case of this concurrent administration, it gets really easy, and it is this steady state level is immediately attained. Equation for the loading dose is steady state concentration multiplied by volume of distribution. If these two values are known, easily the loading dose can be calculated. The suitable loading dose, as we have seen in the previous graph, that there are various loading doses can be administered at various concentration, but no matter how high dose is being given, a steady state level is achieved at the same point almost. There is a suitable loading dose, and that can be easily calculated if these two values are known. Another important significance, pharmacokinetic significance of intravenous infusion administration, which we discussed, mentioned already, is that it is highly desirable for potentially dangerous situations. As by stopping the infusion, plasma drug concentration drops exponentially, means immediately it will drop, and the change in the drug concentration will start to show it in a negative mode. So, which is desirable in such a situation. Now, on the other hand, if you compare or contrast it with oral administration, or so what happens in that situation, that if steady state level is there, um, and the patient starts to experience unwanted, undesirable side effects, toxic effects, and you want to reduce the level, and you stop the administration, it will take a while to reduce the drug concentration from the steady state level. It won't drop immediately. So that is the main advantage which is associated with intravenous administration, and its administration is particularly useful in case of drugs with narrow therapeutic indices, for example digoxin, lithium, and gentamicin. As we know that digoxin, if it is a very potential drug, and it has very narrow therapeutic window, and within no time it can cross the toxic minimum toxic concentration, and its undesirable effects are directly on the heart. So similarly, in case of gentamicin, we can see that crossing the toxic concentration, it might cause nephrotoxicity and permanent kidneys damage, which is extremely undesirable condition or the side effect. So that's why for such drugs it is highly recommended to follow the intravenous infusion pathway. So coming to the graph, this graph we already discussed, and this graph very successfully and very easily explains that why intravenous infusions are so safe. You can see 2 points are there, B and A. B is sufficiently before the steady state level, and A is immediately before the steady state level, but asymptotically it is the same as the steady state level. No matter you are dropping or stopping the intravenous infusion at this point or at the point B, the level will drop exponentially and the concentration will abruptly decrease. And the same is applicable to this point as well. The concentration will drop exponentially and the concentration will start to decrease. So definitely if the levels start to decrease, the patient is now safe, patient is not in danger zone. The concentration which was causing the toxic effect, that is now at its decreasing mode. But in contrast to this one, what we see in the steady state level which we achieve in oral administration, let me explain here. For example, this is a graph which we are obtaining after the oral administration. So as you know that what happens, first dose is given and then the second dose is given, level keeps rising, third dose is given, so on and so forth, and slowly and gradually your level is increasing. So, and for example a point is reached when your steady state level has been attained. So now you can see that now this steady state level and this steady state level is so much different. In this steady state level there are constant fluctuation between the top and the trough levels. There is a top level and then there is a baseline level, and for example as the patient start to witness the toxic effect, what will happen that if you reduce the further oral dose administration, it won't happen that uh, the concentration will drop immediately, like at this point you have stopped. so it will drop immediately like this. No, this won't happen, but rather than the level will stay and fluctuate at the same concentration little by little at the same level, and then it will start to come downward. So you can see that the oral administration is not that safe in this way as compared to the intravenous infusion. This is much more safer, and I hope now it is the difference is now easily clear. So that is the main reason that digoxin, lithium, and gentamicin are usually in critical situations are avoided, because the drugs which got narrow therapeutic indices, if get trapped in such situations that you're giving them orally and the level is not dropping immediately, though the drug has crossed the toxic concentration, it is extremely potentially dangerous situation for the patient, and clinicians want, it is a nightmare for clinicians, they want to avoid it at any cost. So that's why intravenous infusion is so desirable in such situation. Intravenous infusions of two compartment model drugs. Two compartment model drugs, as you know, is would be followed by the drugs which are sufficiently lipophilic. Like one compartment model, in case of one compartment model, steady state level would be achieved rapidly, and the reason is because the, the drug is not that much distributed into the peripheral tissues, so it would take lesser time to be in the to come into the redistribution phase and gain its equilibrium back. So many drugs given by intravenous infusions follow two compartment kinetics. So means when until now we discussed the one compartment model, but it doesn't mean that most of the drugs are following the same. Many drugs are there which follow the two compartment kinetics as well. For example, distribution of theophylline and lidocaine. These are the drugs which got sufficient lipophilicity, and they have this tendency to distribute towards peripheral tissues. So what happens in case of such drugs, that with two compartmental model drugs, intravenous infusions require a distribution and equilibrium of the drug before a stable blood level is reached. Drug first will be distributed to the far areas, and then it will start to come back into the blood circulation, because this is a two directional or reversible process. So that coming back and then first the distribution and then coming back and then attaining the equilibrium, that takes time. So that is the reason that the steady state level, which is associated with the those drugs which are following two compartment model, which in other words means that the drugs which got sufficient lipophilicity, in those situations the steady state level would take time to achieve, and if that is necessary to achieve rapidly, so in that situation the intravenous infusion concurrent administration with a loading dose is the best idea. So here you can see that how the knowledge of kinetics is helping to figure out various clinical situations and dealing with various drugs, nature of various drugs in clinical situations. So during a constant intravenous infusion, the drug in the tissue compartment is in distribution equilibrium with the plasma. Means that here the important thing is that in the drugs which are following two compartmental model, um, there isn't only the absorption and elimination important and dominant phases, but distribution as well. So what happens that when the drug goes into the tissue compartment, that means during the distribution, as the drug will be in the systemic circulation, it will start to be distributed as well. So you can see that the rate of elimination, which is already first following first order and concentration depending, that now it is lacking, because the drug has moved to peripheral tissues and its distribution and equilibrium is taking time, and so the building up of the concentration though it is building up in the tissues, but in the plasma, that is taking time. The distribution and the equilibrium is taking time, and that is the main reason that it takes longer to attain the steady state level in case of the drugs following the two compartmental model. Constant steady state concentration level also result in constant drug concentrations in the tissues. No net change in the amount of drug in the tissue occurs at steady state. Now, this is another issue when, in case of the drugs which are following the steady state level, that it has to be waited for some time to reach the drug or attain the drug its equilibrium back after the distribution phase, because if equilibrium isn't established yet, there is chances that the steady state level, so-called steady state level, might show or exhibit further changes, and because of that there would be change could be witnessed in the steady state. There will be fluctuation in the concentration in the steady state level. So that is the reason that steady state level or the stable steady state level, or which is also called the terminology is used for drugs following two compartmental model, is constant steady state concentration. So avoiding the toxic concentration is another benefit of the knowing the pharmacokinetics of intravenous infusions in clinical situation. By slow infusion rate and low quantities, toxic limits are easy to avoid it. We have discussed it multiple times, and I think it doesn't need any further elaboration or explanation. If the saturation kinetics is reached, stopping them abruptly reduces the toxicity chances. Um, though as we know that saturation kinetics is the one which is in which the elimination rate is also following has also started to follow zero order kinetics. So in that situation, when even if we are dealing with the intravenous infusions, when you will stop the intravenous infusion, what will happen that it will take some time to reducing the intravenous infusion, for because it will wait for the elimination rate to start following again the first order. It comes back from the zero to, and definitely it will take time for the transporter enzymes to come back from the saturation to unsaturation, and so that the elimination rate again start to follow the first order kinetics. So that is these are the various reasons and things which are involved whenever the kinetics of intravenous infusion becomes very extremely important in clinical situations. So toxic concentrations can be avoided, but if that saturation kinetics has started, then it is much easier and possible to attain the non-toxic states through the intravenous infusions and the oral administrations. If the patient is experiencing side effects, administration can be stopped immediately. Okay, students, thanks. This was end of the chapter. I hope that now you people are well aware about the concept of intravenous infusions, and you can easily understand that what are the hallmark characteristics of kinetics of pharmacokinetics of intravenous infusions.